Breakthrough. Plant virus reprograms immune system to fight cancer. What if a virus from a humble bean could outsmart cancer? Researchers at the University of California, San Diego are uncovering how a plant virus, the cowpea mosaic virus, CPMV, might hold the key to reprogramming the immune system to target tumors. Published in Cell Biomaterials, their findings reveal why CPMV sparks a powerful anti-cancer response, while similar viruses fall short. But here's where it gets controversial. This virus doesn't even infect human cells. So how does it teach the immune system to fight cancer? Let's dive into the science and the questions it raises. This study, led by Dr. Nicole Steinmetz and graduate student Avani Omale, compares CPMV to its cousin, the cowpea chlorotic mottle virus, CCMV. Both viruses look alike and are taken up by immune cells, but their effects diverge dramatically. When CPMV is injected into tumors, it acts like a master conductor, orchestrating a symphony of immune activity. Neutrophils, macrophages, natural killer cells, and dendritic cells swarm to the tumor site, while B cells and T cells gear up to create lasting immune memory. This dual action not only attacks existing tumors, but also primes the body to hunt down metastatic cancer cells hiding elsewhere. But what makes CPMV special? The answer lies in its RNA. Unlike CCMV, which triggers inflammatory signals that fail to clear tumors, CPMV's RNA lingers in immune cells and activates toll-like receptor 7, TLR7. This receptor is a critical player in antiviral and anti-tumor defenses. By hijacking TLR7, CPMV essentially tricks the immune system into treating the tumor as an invading pathogen. And this is the part most people miss. CPMV doesn't need to infect cells to work. Instead, it's recognized by pattern recognition receptors, PRRs, as a foreign invader, sparking a chain reaction of immune signals. The results in mice are striking. Tumors injected with CPMV shrank, and even distant, untreated tumors regressed, a sign of a systemic immune response. Even when stripped of its RNA, CPMV's virus-like particles still activated immune cells through TLR2 and TLR4, proving its versatility. In canines with cancer, CPMV showed tumor-agnostic efficacy, inhibiting cancer-promoting signals and boosting long-term immunity. Yet, the team emphasizes that this is just the beginning. While CPMV's safety and effectiveness in humans remain to be tested, the study highlights a bold idea. Nature's pathogens, from plant viruses to bacteriophages, could be repurposed as tools for cancer therapy. As Dr. Steinmetz explains, the immune system is a universal detective, capable of recognizing molecular patterns from viruses across the tree of life. So, what's next? The researchers are refining formulations, dosages, and treatment protocols to prepare for clinical trials. But questions linger. Could CPMV's non-infectious approach be safer than oncolytic viruses which directly kill cancer cells? And how do we balance the risks of immune overactivation with the potential for tumor destruction? The science is clear, but the debate is just starting. Do you think this plant-based strategy could outperform traditional immunotherapies? Share your thoughts in the comments. After all, the future of cancer treatment might just be growing in a bean field. Breakthrough plant virus. How CPMV trains your immune system to kill cancer. What if a virus that infects plants could reprogram your immune system to destroy cancer? This isn't science fiction. It's a groundbreaking reality emerging from labs at UC San Diego. Researchers have discovered that the cowpea mosaic virus, CPMV, a pathogen typically found in black-eyed peas, acts as a powerful ally in the fight against cancer. But here's where it gets controversial. Why does this humble plant virus outperform even advanced human-engineered therapies in triggering the body's defenses? Let's unpack the science and the debate. In a landmark study published in Cell Biomaterials, scientists revealed that injecting CPMV directly into tumors sparks a remarkable immune response. Unlike other plant viruses, CPMV rallies an army of immune cells, neutrophils, macrophages, and natural killer cells, to infiltrate tumors and dismantle cancer cells. Simultaneously, it trains B cells and T cells to remember and hunt down cancer long after treatment ends. This dual action not only shrinks targeted tumors, but also attacks hidden metastatic growths elsewhere in the body, And this is the part most people miss. CPMV's effects aren't temporary. It creates a lasting immune memory that keeps cancer in check. It's astonishing that CPMV, a virus humans can't even catch, reprograms our immune system so effectively, says Nicole Steinmetz, the study's lead researcher. Her team compared CPMV to its cousin, the cowpea chlorotic mottle virus, CCMV, which shares similar traits but fails to combat cancer. The key difference? CPMV activates 3 types of interferons, proteins renowned for their cancer-fighting abilities, while CCMV triggers less effective inflammatory signals. Even more crucially, CPMV's genetic material lingers in immune cells, activating a critical defense pathway, TLR7, that supercharges the body's anti-tumor response. But here's the twist. Could a therapy derived from plants really rival high-tech cancer drugs? Critics argue that plant-based treatments lack the precision of synthetic therapies. Yet CPMV supporters highlight its game-changing advantages. Unlike expensive lab-made drugs, this virus is grown sustainably in plants using sunlight and water, a process called molecular farming. It's affordable, scalable, and surprisingly potent, explains Anthony Omol, the study's first author. We're not just fighting tumors, we're redefining how cancer therapies are made. The team is now pushing toward human trials, but questions linger. Why does CPMV uniquely awaken the immune system? Could its plant origins limit its effectiveness in humans? And here is a thought to debate. If this virus works so well in dogs and mice, should we fast-track its testing in people, despite the unknowns? Share your take. Would you embrace a plant-grown cancer therapy, or does the idea leave you skeptical? This research, funded by the NIH, American Cancer Society, and others, could revolutionize immunotherapy. As Steinmetz puts it, we're at a turning point. The science is clear. Now we need the courage to explore new frontiers. Plant virus CPMV, a breakthrough in cancer immunotherapy. Imagine a tiny virus known for causing trouble in black-eyed peas now holds the key to unlocking a powerful, affordable cancer treatment. This is not just science fiction. It's a reality that's catching the attention of researchers around the globe. In a fascinating study published in Cell Biomaterials, a team led by chemical and nano-engineers at the University of California, San Diego, delved into the unique properties of the cowpea mosaic virus, CPMV, a plant virus that stands out from the crowd. Unlike its counterparts, CPMV has the remarkable ability to activate our immune system to target and destroy cancer cells. The study, titled Comparative Analyses for Plant Virus-Based Cancer Immunotherapy Drug Development, reveals how CPMV, when injected directly into tumors, recruits a battalion of immune cells, neutrophils, macrophages, and natural killer cells to launch a full-scale assault on cancer cells. This is just the beginning. The virus also awakens B cells and T cells, creating a lasting memory that primes the immune system to hunt down cancer cells throughout the body. Nicole Steinmetz, the study's corresponding author, marvels at the selective power of CPMV. It is fascinating that CPMV, but not other plant viruses, stimulates an anti-tumor response. She says, but how does this work? According to Anthony Omol, the study's first author, human immune cells, despite not being infected by CPMV, respond to it and are reprogrammed to an activated state. What we found most exciting is that although human immune cells are not infected by CPMV, they respond to it and are reprogrammed toward an activated state, which ultimately trains them to detect and eradicate cancerous cells. The real question is, what makes CPMV so effective against cancer? To find out, Omel and Steinmetz compared CPMV with the cowpea chlorotic mottle virus, CCMV, a closely related plant virus that lacks anti-tumor effects. Despite similar appearances and uptake rates, the viruses produce vastly different outcomes. CPMV stimulates interferons, proteins known for their anti-cancer properties, while CCMV triggers pro-inflammatory interleukins that fail to clear tumors effectively. The RNA processing of CPMV also plays a crucial role, as it persists longer and activates toll-like receptor 7, TLR7, a key player in priming anti-tumor immune responses. But here's where it gets controversial. While many cancer immunotherapies require complex and costly manufacturing, CPMV offers a cost-effective alternative. It can be grown in plants using sunlight, soil, and water, explains Omo. The team is now working to advance CPMV to clinical trials, with Steinmetz noting, "We are diligently working toward the next steps to ensure that the most potent lead candidate is selected to achieve anti-tumor efficacy and safety." As we stand on the brink of a new era in cancer treatment, it's worth asking, could a humble plant virus be the key to unlocking the full potential of our immune system against cancer? Let us know your thoughts in the comments. Reference: Anthony Omo et al. Comparative Analyses for Plant Virus-Based Cancer Immunotherapy Drug Development. Cell Biomaterials 2025. DOI.10.1016.seedlubtear.cellbio.2025.10095.